So today we're going to be talking about fatty acid synthesis. This is a process that happens in the body where we form long-chain fatty acids from acetyl-CoA molecules. So you'll notice the acetyl-CoA molecule only has 2 carbons, and this long-chain fatty acid is called palmitate, and it is 16 carbons long. So it would take 8 acetyl-CoA molecules stitched together to make our big long-chain fatty acid molecule. So why do we do this? What are the purposes? So number one is to store energy. So if you've ever heard that energy is stored in hydrocarbon bonds, that's what's happening here. We have a bunch of little HC bonds that store all our energy here in the form of these long-chain fatty acids. Another purpose, when we synthesize fats, this can help us remove sugar from the blood. And we'll try to look a little more about how that happens, but sometimes building fats is a way to effectively lower your blood sugar right after a meal. All right, so where does this happen? So in the body, when we're talking about energy and the management of sugars, fats, and those levels in the body, we should always be thinking of the liver. This is the general control center for those levels in the body, right? So liver cells are going to be the site of our production of fatty acids. This can also happen in adipocytes or our fat cells that store those fat droplets, but this is going to be a little bit less than liver cells. Okay, so where in these cells does it happen? That's very important. So in the cells, fatty acid synthesis occurs in the cytoplasm of the cell. Okay, so let's take kind of more of an in-depth look, just a map of what's going on with our molecules here. So I've got a liver cell with its mitochondrion, and we're in the postprandial state. And they'll use this term a lot, but it just means we just had a big meal, okay? We just had a big meal. So let's say, it's like a big burger or something. Lots of sugar in this, right? And that bread, amino acids in the meat. But we're mainly going to look at the consequences of the sugar from this, right? So a big meal is going to be absorbed through the intestine, and we're going to get a lot of glucose in the blood, okay? And since the liver is the main control center for glucose transporters are going to bring in a lot of that sugar. And we know after a meal, our insulin is going to spike, and this is going to help with that response of pulling glucose into the liver cell, okay? So insulin increases this process. Now, what do we often do with glucose when there's an excess of it? We run glycolysis, right? We break down those glucose molecules into pyruvate, okay? And insulin is also going to increase this process, glycolysis, right? Let me move over our mitochondria so we have a little bit more room here. Okay. So now that we have pyruvate, we can shuttle this into the mitochondria. Okay, so pyruvate is now in the mitochondria, and we know this is where we make most of our energy, and that pyruvate can be converted to acetyl-CoA, which we talked about at the beginning of the video, by an enzyme called pyruvate dehydrogenase complex. Okay, so we convert our pyruvate to acetyl-CoA via the pyruvate dehydrogenase complex. Okay, so now that we have acetyl-CoA, there's a couple things we can do with it. Number one being we can take it to the citric acid cycle, and this makes ATP. That's one thing. Draw that in red for our energy. So it makes ATP in the form of GTP, which is then converted to ATP. And it also makes electron carriers. And we then use those in oxidative phosphorylation. Getting a little jumbled, sorry. But that makes more ATP, okay? So after a meal, we've got enough sugar in the body, which means we've also got enough ATP, right? It's formed in glycolysis, it's formed in citric acid cycle, it's formed in oxidative phosphorylation. So in the postprandial state, we're not really, we don't need a lot of ATP made. We don't need to ramp up our super energy making processes. So that is where fatty acid synthesis comes in. This acetyl-CoA has to go somewhere else if it's not going to the energy creating processes. So what's going to happen with it is it's got to go out into the cytoplasm for the fatty acid synthesis, right? So we use something called the citrate shuttle to get the acetyl-CoA back out into the cytoplasm. Okay, we'll go in depth later in the video about how the citrate shuttle works. But now that we have acetyl-CoA out in the cytoplasm, we can take that to fatty acid synthesis, which is going to produce long chain fats, like our palmitate that we talked about initially. Okay, so I want to go a little bit more in depth with the citrate shuttle because I think it can be commonly tested and really misunderstood. So let's talk about what's happening. So inside the mitochondria, our pyruvate is converted into the acetyl-CoA via the PDH complex, that pyruvate dehydrogenase complex, right? So we've got a lot of cofactors involved there, but the main deal is we get acetyl-CoA out, right? So in the citric acid cycle, we know that acetyl-CoA combines with oxaloacetate to form citrate. And if this doesn't seem familiar, it's part of the citric acid cycle, and acetyl-CoA is a 2-carbon molecule, and oxaloacetate is a 4-carbon molecule. So that means that adding these together, we'll get a 6-carbon citrate molecule, right? So the important thing about citrate is it's able to pass through this mitochondrial membrane to the cytoplasm. So this is what we mean when we're talking about the citrate shuttle, is we're using citrate as a kind of intermediate molecule that's able to pass through the membrane. So once citrate passes through the membrane, it can be converted back into acetyl-CoA and oxaloacetate. So I'll draw 2 lines here. So oxaloacetate is produced again. It's that 4-carbon molecule. And then we have acetyl-CoA left over. Okay, so let's think about what went into this process and what went out. So we input acetyl-CoA on the mitochondrial side, and we output acetyl-CoA on the cytoplasm side. Citrate is our intermediate molecule. So this is the citrate shuttling of acetyl-CoA to the cytoplasm from the mitochondria. All right, so now we have the acetyl-CoA in the cytoplasm ready to use. But one thing to understand is that acetyl-CoA is not a very active molecule when it comes to fatty acid synthesis. You don't just put it out in the cytoplasm and it turns into a big fat. You have to have some other things occurring to get it to that state. So the most important activating step is the conversion of acetyl-CoA to malonyl-CoA. Some people say malonyl, same thing, right? So acetyl-CoA and malonyl-CoA, the only difference between them is acetyl-CoA is a 2-carbon molecule and malonyl-CoA is a 3-carbon molecule, okay? And if you're noticing a pattern with some of the molecule transformations in the citric acid cycle, when we have a difference in the number of carbons, the main thing that's going to be exchanged is a CO2 molecule. So we add one CO2 molecule to get the extra carbon into malonyl-CoA from acetyl-CoA. And one thing I want to highlight is, like we were talking about, malonyl-CoA is a very active molecule. Once we get to this step, it's like we're committing to fatty acid synthesis. So if you have malonyl-CoA in the cytoplasm, that's getting transformed into a fat. Nothing you can do about it at that point. Okay? So the enzyme that regulates this step is called acetyl-CoA carboxylase. All right. And that's kind of intuitive, right? Because we're inserting CO2 onto the molecule, or stitching it onto the molecule. And if you remember from other cycles, biotin is an integral part of transferring CO2 groups within enzymes. So the biotin is that long arm structure that kind of takes CO2 and moves it over here and, smacks it onto a molecule, simply put. So biotin is going to be required. So the MCAT loves to test on this. It says, if there's a deficiency in biotin, would we see increased or decreased fatty acid synthesis? It's going to be decreased because it decreases the activity of that activating step where we get malonyl-CoA out of acetyl-CoA. All right? So now we have our activated molecule, malonyl-CoA. What happens to it? So the main enzyme in fatty acid synthesis is namely fatty acid synthase, right? So we just tack on the ACE to the end there. So fatty acid synthase is a giant enzyme, and it makes use of something called substrate tunneling. Not super important for answering all the questions, but just know that the enzyme is really big and has a lot of active sites that kind of push molecules together and change the environment so that we can stitch together a bunch of small molecules to make a big long-chain fatty acid, right? So what cofactors are involved here? So the number one thing that you need to worry about is NADPH. This cofactor is required for fatty acid synthesis, right? So NADPH. And what is NADPH? It is an electron carrier. So why is this important? So malonyl-CoA is a very oxidized molecule. So if I'm to draw it out here, so we've got that S and then CoA group there, and you'll see these carbons are heavily oxidized by these oxygens here, right? So that means we've kind of used all the energy that they've got, and that's kind of a trend we'll see with other molecules. Pyruvate is more oxidized than glucose, acetyl-CoA more oxidized than glucose as well. So as we get energy out of the molecules, they become more oxidized. NADPH carries electrons over and helps to reduce these molecules, right? To create energy and store energy, we have to reduce these oxidized carbons to have those lots of HC bonds to store the energy, right? So cycles are going to occur in this fatty acid synthase where we add on more and more acetyl-CoA molecules, and we use more and more NADPH. So this will happen lots of times, and we're finally going to make that palmitate that we had at the end. So that super long chain fatty acid that has 16 carbons, right? So all of those carbons like this, these are super reduced, the best you can get. So that's why they store so much energy. All right, let's talk about some of the regulation of this fatty acid synthesis process. So I've kind of got a general summary of what's happening here. We have citrate, oxaloacetate, and acetyl-CoA there, and then going to malonyl-CoA, and finally palmitate, our final product, right? So what hormones are going to accelerate this process? So remember, we talked about one of the purposes being to remove sugar from the blood, decrease that blood glucose after a big meal, and the hormone that has the most to do with that is going to be insulin. So you can expect these enzymes that convert those sugar molecules from glucose to citrate to acetyl-CoA, all of that, converting it to fatty acids, you would expect insulin to increase the activity of these enzymes. And if you think about it, it makes a lot of sense, right? We want to remove sugar from the blood. That sugar has to go somewhere. So we're using glucose as kind of the building blocks, right? When we break it down into acetyl-CoA and other molecules and we build fats, we're removing sugar from the blood, and that's one of insulin's main purposes, right? Also, we kind of have a feedback mechanism or a feed-forward mechanism here where citrate, if there's a buildup of that, will also increase the activity of these enzymes, right? So if we have too much citrate, remember citrate goes through the tricarboxylic acid or the citric acid cycle to produce ATP and electron carriers, which go to oxidative phosphorylation to make more ATP. So if we have really high levels of citrate, we probably also have really high levels of ATP, right? So this high levels of ATP will negatively feed back on these processes that produce more ATP, right? So if we're not sending our citrate to the tricarboxylic acid cycle, it's got to go somewhere else. So we have to ramp up these enzymes to produce fatty acids with our citrate molecules and eventually acetyl-CoA molecules. Okay, so now let's talk about some hormones that would inhibit the fatty acid synthesis process. Number one is going to be glucagon, okay? So glucagon, epinephrine, norepinephrine, these hormones are all about raising the blood sugar, raising the blood glucose level so that, maybe we're really hungry, we need to get some glucose back in the blood, or, it's a fight or flight situation where it's like, get glucose in the blood now so I can run away from this thing or I can fight this thing. So if we want to put sugar back in the blood, that's kind of the opposite of storing energy as big fatty acids for like a long-term storage facility. Okay, so glucagon is going to inhibit these enzymes. Okay, and you can kind of think about this as if we don't have enough glucose, we're not going to be using glucose to store energy, right? We want to produce more glucose, so we've got to go the other way around, right? So showing that with the bottom version as well, if we have a really low amount of ATP, then all of our citrate and all of our acetyl-CoA needs to be fed into the tricarboxylic acid cycle, and then those electron carriers need to be fed into oxidative phosphorylation because we need more ATP. So we aren't going to be making fats. All right, thanks for watching, and do let me know if you have any questions below. I'll do my best to answer them, so just leave them in the comment section.